So we're just talking about active transport needing energy, and we're going to carry on talking about energy and the cell and ATP specifically here as a molecule. So that's what's coming up. And then we're going to round this out by talking about enzymes in this second video. So it doesn't matter how tired you are right now, how sleepy, how lazy you feel, how cozy you are under a blanket. On a cellular level, there's lots going on. They're always busy and are working. If we think about that cellular level, there are thousands of chemical reactions going on in that cell. And what it does is it keeps the cell alive, it makes new cell parts, and ultimately can help make new cells or have cell duplication. And so if we were to talk about these thousands of chemical reactions, some of them release energy or are exergonic, and some of those chemical reactions require energy or are endergonic. And so we're going to talk about those two kinds of energy. And we're also going to talk about energy coupling in the cell and how that can really help us understand how cells stay alive. So before we get into that, a little bit of physics and biology, just for fair play here. I've been talking so much about chemistry and biology. How about a little bit of physics? So if we were to define energy, it's the ability to do work or the capacity to cause change or to perform work. There's two different kinds. So the energy of motion or energy of movement is kinetic energy. And we'll talk about how heat energy is related to kinetic energy. And there's also stored energy or potential energy. Sometimes we talk about gravitational potential energy. When something's at the top of the hill, it has potential energy as it comes down or to come down. And so we're going to talk about heat, and we're also going to talk about chemical energy and potential energy. And so we're pretty lucky to live on a river in this area, or in the very least live near hydroelectric dams. And so both hydroelectric dams illustrate both of these kinds of energy. So kinetic energy, or that movement, is where water is moving through a turbine to create energy. So oftentimes we see energy transformations as something. And this is an example of kinetic energy because there's motion, right? It's moving. Heat or thermal energy is kinetic energy if we move down to the molecular level. And so if we were to move into a molecular level, if you have a cup of water on your desk right now that's cold, the molecules, maybe not atoms, are moving. But if you had a cup of tea on your desk right now, the molecules would be moving much faster because there's more heat energy. And so that speed of movement is akin to heat. And that's why heat is considered a kinetic energy. You have to zoom way in to that molecular level. Going back to the hydroelectric dam connection to these two kinds of energy. Potential energy is stored energy. So the water being stored behind a dam is an example of potential energy. And chemical energy, when we talk about the energy that's stored in chemical bonds, that is potential energy because it could be released in a chemical reaction. Chemical reactions involve breaking and making those chemical bonds, or making and breaking those chemical bonds in either case. So the idea here is that when you break down molecules like glucose, you can release the energy in that. And that's what we do with our digestive system, but it's also what we do on a cellular level in the process of cellular respiration. So the glucose had energy before it was converted into energy and carbon dioxide and water. So a few laws of the universe here, just to ground our discussion of energy and transformations of matter, chemical reactions, or biochemistry. First law of thermodynamics says that energy isn't created or destroyed. It is recycled or converted from one form to another. So there's might be a loss of energy, but it goes somewhere. So in the case of an exergonic reaction, the energy is released to the atmosphere, but it's still accounted for in the universe. And so we can't create or destroy energy. but we can convert it from one form to another. The second law of thermodynamics explains why your apartment gets messy or your purse needs to be cleaned out at the end of the week, or why your kids' backpacks are a disaster. Entropy is the measure of disorder, or I like the word chaos, in the universe. And there is a law of the universe that things tend towards chaos or tend towards disorder. And so this idea that a chemical reaction that moves downhill, like this exergonic one that I was just drawing here, we'll come back to those drawings. Maybe they'll make a little more sense. These downhill chemical reactions can happen because they have more and more disorder. And so we've got order up here and chaos down here. Whoops, chaos. And you can attest that it takes energy to put things back into order. If we're talking about your apartment or your purse or your kid's backpack. So this law, second law of thermodynamics, is to explain why you need to clean your house on the weekends or on Monday when you start to do your homework or whatever it is. The world tends towards chaos, and chemical reactions are not outside of that law. Okay, so the two kinds of chemical reactions would be endergonic and exergonic. We're going to start with the exergonic, so ergon for energy, and the energy is exiting. So the exiting energy means that we start off with reactants with a lot of energy and we create products with a small amount of energy. And this energy is maintained. Remember, we can't create or destroy energy, but it is lost when we take a look at the energy levels of the product. So the examples of this would be burning wood or burning of anything. So we've got wood up here with lots of energy, and it's going to combine with oxygen to produce our ash, carbon dioxide, and maybe a bit of water, right? But the key thing is that that's producing heat energy for my house, right? That would be an exergonic reaction. If we were to go with, a more biological explanation here, we could talk about cellular respiration. And so cellular respiration is also exergonic. So we start out with something with a lot of potential energy in it. In the case of cellular respiration, that's glucose. Also combining with some oxygen that we breathe in, and it's going to produce a whole lot of energy for our cells to use, but it's also going to produce CO2 and water. In these little molecules, there's way less bonding, way less things combined in energy packets. And so we've got way less energy in the products than we had in the reactants. And this would be a downhill chemical reaction that illustrates an exergonic reaction because energy is coming out of that chemical reaction. The reverse of an exergonic reaction would be an endergonic reaction. Oh, sorry, I forgot the two pictures here. I've already kind of drawn them out. So instead of wood burning, it was the combustion of a car, a combustion engine. And so it illustrates that. And they really are similar, right? They both put energy out. In the case of heat energy or movement energy in this car, or heat energy and ATP in the case of cellular respiration. Sorry, now the reverse of this. So the reverse of an exergonic reaction is an endergonic reaction. So the energy has to go into, so it has to enter, get it? Enter. I don't know if that works for you. So it requires an input of energy. And we start with reactants with a low amount of energy, and we end up with products with a high amount of energy. So the reverse of cellular respiration is photosynthesis. And I chose this one because it is something that we've talked about. Now, your cells don't have photosynthesis going on in them, but it's a good example. So the energy comes from the sun, and we put carbon dioxide and water, those low energy reactants, together with the energy of the sun to make glucose and oxygen. And so that energy is accounted for because it goes in and is now stored energy. It's potential energy in the products. So like I said, we don't have photosynthesis going on in our cells, but we do have endergonic and exergonic reactions going on. And the sum total of that is your cell metabolism. So hopefully it balances out and keeps your cell alive and doing the things that are vital for your cells. So what it gets used for, what that energy gets used for in the exergonic reactions would be chemical work. So driving endergonic reactions, that energy coupling that we're going to talk about more and more. Also, we're going to talk about how all chemical reactions actually require a buy-in or an energy of activation. Mechanical work, so it could be the beating of cilia or a flagella, any kind of locomotion or movement. Transport work we've talked a lot about, so any active transport. So it could be a sodium potassium pump, it could be endocytosis or exocytosis. Those would all be active transport on a cellular level. So to accomplish this work, a cell has to manage its energy resources, and it's this energy coupling that I've said already. We've got the exergonic processes driving the endergonic one. So we have energy being produced and energy being used. And the energy that we're talking about is ATP, and ATP drives all three types of work. That is the essential molecule for energy in our cells. Oftentimes it's said that ATP is the currency of the cell. It's what you have to spend in order for things to happen. And so if we were to take your whole body, and I don't recommend doing this, but separate it out into all the molecules that make you up or make up you, ATP would only be 50 grams of it. So you consider all the work that has to be done to keep your body alive, all the work that has to be done for you to do what you need to do in a day. And we only have 50 grams. That's a tiny amount of ATP. And so why we have or can have such a small amount is because we have this constant recycling of ATP. It's a renewable resource so long as we can recharge it. So the source of energy for constructing ATP is food. So you digest food and take that potential energy and then store it into ATP. And if we were to think one step back from there, the sun actually grew the food you were eating so that you could eat it and then make the ATP. And if you don't eat the plants, then you eat an animal that ate the plants. And so ultimately the root of all this is the sun. And so photosynthesis does come into this, and there is a chain of energy through to the ATP molecule. An ATP molecule is simply the carrier and storage unit. It's the way our cells package the energy so that it's in a useful form. So what ATP is adenosine triphosphate. And so we're going to have three parts of this adenosine triphosphate. We have an adenine, which is nitrogenous base. We have a ribose, a 5-carbon sugar, and then we have 3 phosphate groups. Says that there is a high amount of energy held in some of its bonds, and specifically between the second and third phosphate groups here. I grew up with 2 sisters, and we used to have to share 3 to a seat on the bus, and I have to say there was often a lot of energy between the sisters. Maybe it wasn't always good. But if we were to, on a molecular level, break this bond apart here, we could hydrolyze or hydrolysis of ATP. Remember that takes water and puts it in to break it up so that we take that phosphate off, or the third sister leaves, that is going to release some energy. So we release the energy and a phosphate group. Instead of ATP now with 3 phosphates, we now have ADP with 2 phosphates. So adenosine diphosphate down here. So let's take a look at a picture here, that idea of going, or what ATP is and how it goes to ATP or ADP. My goodness. So we have adenosine as the nitrogenous base. We have the 5-carbon sugar ribose, and then the 3 phosphate groups. If we were to put water into that molecule, we could break apart the phosphates so that we break one phosphate off. When you break that bond, you're going to release the energy. You're going to release the potential energy that was stored in that covalent bond. And that's really what this is after. So that's how the ATP was storing the energy. So now instead of 3 phosphates, we have 2. And this would now be, this molecule here, would now be adenosine diphosphate. ATP becomes ADP, and what that energy is used for is all the stuff that I was talking about before. So chemical work, driving chemical processes, even the downhill ones, even the exergonic reactions require some energy to go into them. Mechanical work, so the contraction of muscles, it could be anything for locomotion on a cellular level. And then again, that transport work, any active transport requires energy to move things against the concentration gradient. Going back to that idea that there's only 50 grams of ATP in your body, it does have to be renewable. And so how it gets renewed is that we just talked about how ATP becomes ADP and releases energy. Well, that energy is for driving endergonic reactions. We talked about how that process was excuse me, hydrolysis. Well, if we were to recharge that ADP and phosphorylate it, put 1 phosphate back on, we can do that if we have reactions in the cell that release energy. So it releases energy to put that phosphate back on the ADP and recharge our source of ATP. So when energy is released in exergonic reactions, such as the breakdown of glucose, and the energy is then used to regenerate the ATP. So this energy coupling and the renewable nature of ATP are really important for your cells to stay alive. Along with the chemical reactions and energy conversation, enzymes factor prominently in this. So we know that enzymes are proteins. Remember, that's your default answer. And we have several variations of definitions of what enzymes are. So we're going to start with the first one here. And I think there's about 3 different ways of saying this. So the first one is that enzymes speed up cells' chemical reactions by lowering energy barriers. There's always a buy-in, even for these chemical reactions that could happen spontaneously, these exergonic reactions. It takes money to make money. And so there's that idea of a buy-in or an energy barrier that the chemical reaction has to climb over in order to get to where it's going energetically. So there's a lot of potential energy in biological molecules. So this idea that you can't just release it spontaneously. Energy has to be available to break the bonds so then new ones can be formed. And that energy that has to be available to buy in is called the energy of activation, sometimes called E of A in a shorthand way in chemistry. The other definitions here, so we said that they are used and speed up chemical reactions. We could also say that they are catalysts. And a catalyst is a driver or a sparker to biological reactions. This sparking we're talking about is what the enzymes are doing. So enzymes can speed up the energy or speed up the rate of the reaction by lowering that energy of activation. They are not used up in the process. They are another renewable thing. So if we were to talk more about enzymes, each enzyme has a very particular target molecule. We call that the substrate. And as my example here, most of us understand that lactose is an enzyme in our digestive system. So I'm going to draw a ridiculous picture of lactose. It does not look like this. And it's going to fit lactase. Lactase is the enzyme. Lactose, I'm sure somebody else was just thinking that, is the sugar molecule in milk. And so lactose actually fits into the molecule of lactase in your digestive system. We would call this the enzyme, even though I got it backwards, and lactose is the substrate. It could also be the reactant in the chemical reaction here. This picture I like a lot because it shows the path, the hill that the chemical reactants have to climb over without the enzymes and with the enzymes. You can see it's substantially lower with the action of an enzyme. And so the reactants and the products end up in the same place, right? The net change is the same, but it's much easier. And when it's much easier, the chemical reaction can happen faster with the enzyme. And so that's what we're talking about with this barrier, this idea of a hill, a buy-in that chemical reactions have to make to have happen. We know from our conversation in chapter 3 that shape is everything with proteins. Remember how we talked about how many levels we can take a look at the shape of proteins? Remember they had funny names like primary, secondary, tertiary, and quaternary. So shape is everything. We talked about how enzymes can be denatured. If they change their shape, they don't work. So shape is critical to their role as a biological catalyst. It's because of the shape that the enzyme has an active site where the enzyme interacts with the enzyme substrate. So that's what I was trying to draw with that ridiculous picture of lactase and lactose. And again, that's nothing like lactose looks like, but it does fit this idea that this would be the active site of the enzyme, and this is the substrate. It's the thing that the enzyme works on. So consequently, the substrate's chemistry is altered to form a product in the enzyme reaction. If we have extreme conditions, so we talked about that before, with pH or heat, you can denature an enzyme. You can denature any protein, but denaturing an enzyme specifically changes the shape of the protein and changes its active site, making it unable to work. And so you wouldn't have whatever chemical reaction it was running happen as easily. I really like this picture because it's kind of what I've been trying to draw, but it does a much better job. So this idea, it's changed. So sucrase is going to be acting on sucrose. So my enzyme is sucrase, and it does have an active site that you can see here, right? The shape. And this shape fits the substrate. So the substrate can fit into the active site. We talk about it binding with an induced fit, or it just fits really what we're talking about. The classic example of a hydrolysis reaction where water comes in to break those two molecules apart. So we're going from a disaccharide to two monosaccharide. So the substrate is converted to products in this step, and specifically with the case of sucrase, but generally with any substrate to product. The products are released, so in this case, two monosaccharides. And look at this, it was in the notes, this idea that it's a cycle. The enzyme is ready to go again. It's ready for action again. It's not consumed in the process. And that was a point that was on the notes, but I really think that this diagram helps illustrate that. So we have enzyme with its active site, we have substrate, we have the idea that they fit, hydrolysis in this case, but the products are released, and it's a recycled thing in that or a cycled thing in that the enzyme is ready to go again. Each enzyme has optimal conditions. So we talked about how extreme conditions can denature it. The 3D shape of an enzyme is affected by its environment. So optimal conditions give the best function of the enzymes. We have different optimal conditions. So you can think about enzymes that might work in your stomach are going to be very good at working at a pH of about 2, right, in your stomach. Now, if you have enzymes that work in your mouth, you don't want a pH of 2 in your mouth. So you have a different pH and different enzymes that work in your mouth. And so we do have a variety of conditions throughout the human body. And this specificity or the optimal conditions for different enzymes can be really different. Temperature can be another one, where an optimal temperature could be for different enzymes. The optimal pH for most enzymes is around neutral, but you can see it's not hard to find exceptions to that optimal condition. So different enzymes, different conditions are for optimal. Enzymes can be produced in inactive forms and called proenzymes. Some enzymes may require non-protein helpers, so cofactors such as these metals or minerals here. Coenzymes such as vitamins, And so it's not always just the enzyme, but it can be these other helpers, if you will. And cells have to control enzyme activity. More is not necessarily better. So your genetics can actually turn on and off enzymes and can make for specific enzymes at specific times. You regulate enzymes through inhibition, and we're going to talk a little bit about inhibition. So a chemical that interferes with enzyme activity is an inhibitor. And this idea that enzyme activity is all good is not always true. So many beneficial drugs work as inhibitors on different kinds of cells. So anti-inflammatories can work like this. Antibiotics work like this. They're specific on a particular kind of molecule in bacteria that are not in you. And so, you know, we can exploit the weaknesses of uh, uh, of enzymes and control and keep ourselves healthy. So some pesticides and poisons are also used for nasty reasons and are also examples of chemical or enzyme inhibitors. Down here, we could talk about some examples. We all know that antifreeze is really bad for dogs, and yet they like to drink it, or cats. And the example for this and the explanation why it's really, really bad for them is competitive inhibition on key enzymes that regulate, you know, these cellular functions that keep cells alive. So competitive inhibition means that there can be molecules that seem really, really close to the shape of the substrate, and they can get into the active site and block it. And so it's competing with the substrate so that the enzyme can't really work as well. So antifreeze, alcohol, all work on this level. Some anti-cancer drugs, so it's not all poisons and bad. If we can interfere with DNA production and cell division, we can keep cancer from reproducing. We can keep cancerous cells from reproducing. And so sometimes this inhibition is good. It's not always for poisons and bad things. So we could have competitive inhibition in the active site, and that's what this is illustrating down here. So we've got competitive, so a molecule that almost fits in the active site blocks the substrate from getting in. We could also have non-competitive inhibition. So this is a molecule that binds with the protein at some spot other than the active site, but it shifts the shape of the protein so that it makes the active site no longer possible for the substrate to fit into. And that's what we're talking about, non-competitive inhibition. So this would be the normal binding site. This would be the good one. And these would be two ways that we could have inhibition. We could have competitive inhibition or non-competitive inhibition. So on top of molecules that are inhibitors, we also have other inhibition called feedback inhibition. And so regulating cell metabolism is important. We don't want just enzymes working out of control. So often the product of a metabolic pathway actually feeds back and inhibits one of the enzymes in the pathway. So this is feedback inhibition. What I'm trying to say here is the more product formed, the greater the inhibition. And it shuts down that chemical reaction. So it regulates that chemical reaction or the pathway. There's a little bit of a preview here in the last few minutes about chapter 6. So I'm just going to go through it. So I have talked a little bit about respiration and photosynthesis. We are not covering chapter 7. So after we're done chapter 6, we move on to chapter 8 because we don't have photosynthesis in human bodies. So this is the chemical reaction for respiration up here. Photosynthesis is the reverse. So it would be 6 carbon dioxide and 6 H2O plus some energy from the sun, making glucose and oxygen. And if you're not one that likes the chemical formulas, you can write it out. Don't worry about the numbers out front. Remember, I've said those are just to balance the chemical reaction because we don't want energy created or destroyed, and we don't want matter created or destroyed. So if you're not a chemical symbol person, don't write it out as chemical symbols. Those are really just the shortcuts when you get tired of writing out the words. So the words are absolutely fine. So we've got carbon dioxide, water, glucose, and oxygen. You're going to have some pre-reading with chapter 6 when you get there after the next lab. And this slide I put in to help you out on that. So chapter 6 is on cellular respiration. The video at the beginning slides here, so I said it goes through ATP, but then it gets into respiration. So that's also a good preview. There are 3 stages in cellular respiration. We're going to talk about glycolysis, the Krebs cycle or citric acid cycle, and the third one. The third one is not just the electron transport chain, but it is this massive pair of words here. It is oxidative phosphorylation. So oxidative phosphorylation has the electron transport chain, but it also has chemiosmosis. And chemiosmosis is where you make the bulk of the ATP or the bulk of the energy in cellular respiration. So it is important to get that third stage, kind of the seed planted properly, as oxidative phosphorylation. So there's a few little notes here, but that's really just to give you a preview into the chapter 6 as we finish up this chapter 5.